We talked a little bit about why we would want to look at the family interacting proteins instead of just Rab11 directly, because if we can tackle something that is an intermediary, we can prevent compromise of Rab11 function in healthy cells potentially. So first we wanted to see, okay, like which ones of these are actually involved. I gave you a little bit of a sneak peek of that when I went over the abstract, but you can see that we found that family interacting protein 2 and 3 are going to be ones where we're seeing a much larger change in immune recognition. Now, NTC, anywhere I say that, that's talking about the control, right? That does not, so that's just going to be what I'm comparing to. And then if you see anywhere I have OT1, OT2, right? These are looking at specifically, like our OT1 cells are the ones that are the T cells that are responsive to this infecle that I mentioned, the really immunogenic um, protein that's being presented in our model, because that's going to help ensure that there is the interaction between that lock and key. So, so this is just showing that we do have changes in that immune recognition when we did a screen for that. And so once you get T cells recognizing their targets, right, once the lock and key fit, you're going to increase the number of T cells, right? They're readying the body to respond to whatever kind of threat. And so that's this is sort of a readout of that. So we looked at what happens if we knock out that gene. Maybe you're familiar with the idea of CRISPR, but essentially think of it as going in with scissors, cutting out, our gene of interest there. And so we did that with
I'm just gonna pause you real quick. Does everybody have sort of a basic understanding of the role of T cells in cancer? I know we talked a little bit about immunotherapy, but
I would love it if someone would like say a little bit more about that because I'm pretty new with all of this.
yeah, you've got a really great schematic there, Oli. If you could maybe just talk them through that a little bit.
Yeah, absolutely. So, when you have a cancer cell that is presenting certain proteins on your MHC molecules that I was talking about before, right? That's going to then interact with these dendritic cells, which basically those are going to what's the best way to describe it? It's like,
Eat little bits.
yeah, essentially, they're going to take up some of that and they're going to process that and present it to those T cells. And the T cells then become primed, which means they are now ready to actually interact with cells. If they see that little bit again, then they will be able to recognize that, right? Before that, they're considered immature. They don't really know what they're doing yet. They haven't been to school. And the dendritic cells get to teach them, which things they should be looking out for. And so
They're like gonna come in and fight, like they're primed to fight and do something.
some of them may be, some of them may be causing other downstream effects. There's a variety of different T cell types. But yeah, the is eventually the idea is that your T cells are going to help combat the tumor.
Could
And
I just pause for a second? Oh, Bob has a question. Go ahead, Bob, and I'll ask mine next.
Sure, I was just going to ask, could this be useful for any type of brain metastasis, or is this specific to breast cancer metastasis to the brain?
I'm not actually sure if there have been other screens for this in other types of brain metastases. I'd have to get back to you on that. I don't know.
Actually, we shared in the email that I sent to you prior to our grasp session, we have each other's contact information. And maybe we could reach out after the GRASP session and find that information and share it with the advocates that are here today.
Yeah, I'd be happy to.